Hello, this is Barry Moran from Trinity College Dublin welcoming you to this Bite-Size Bio web seminar. Today's presentation is entitled Cell Cycle Analysis by Flow Cytometry and is being presented by Derek Davies from Cancer Research UK. Derek runs a large core flow cytometry facility and has a great deal of experience in all aspects of flow cytometry and cell sorting. He is currently Chair of Flow Cytometry UK, Head of ISAC's Shared Resource Laboratory Task Force, and sits on the Cytometry Committee of the Royal Microscopical Society. He also teaches on several national and international flow courses. As always, we'll have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Derek at the end. So now, over to you, Derek, for the presentation.
Thank you, Barry, and good afternoon, good evening, or good morning, depending on where you are in the world. As Barry said, I run a large core facility of cell cycle analysis by flow cytometry, and this is part of the flow cytometry series of the Bite-Size Bio webinar series. So what I want to try to cover in the next half an hour or so is why do we actually want to analyse the cell cycle in the first place, and why flow cytometry is a good way to go about doing that. So as part of that, we'll talk about some of the fluorescent DNA binding dyes that we can use in our flow cytometers. But rather than it being a very didactic presentation about how a flow cytometer works and the science behind it, I thought I'd try and concentrate on some of the missing factors that sometimes don't always come across in online or written protocols. Those factors that are going to affect the success or failure of your cell cycle analysis on your flow cytometer. Some of those factors are going to be related to your samples, how you prepare them, how you fix them, the type of dye that you use. And some of them are going to be related to the cytometer, making sure that your cytometer is optimally set up to give you the optimal results. And there are certain things that you have to take into consideration to make sure that works okay. And towards the end of the webinar, I'll talk a little bit about some of the practicalities of how we run a cell cycle experiment on our flow cytometer. The cell cycle analysis by flow cytometry is not new. It's been around since the dawn of flow cytometry. In the 60s, cell cycle analysis was one of the very first applications which flow cytometry was put. And I just pulled out a screenshot of one of the earlier papers. This is 40 years old, but even by that time, cell cycle analysis on flow cytometers was relatively widespread. Back in the day, this is the machine that I first started on, a thing called a fax analyzer, not even connected to a computer, just connected to a channel analyzer. But it produced plots that are recognisably a DNA histogram. And of course, one of the great advantages, as those of you who were on the webinar yesterday will know about flow cytometry, is that it's a method that allows us to get lots of data relatively quickly, which gives us very good and robust statistics. So and since then, of course, DNA analysis in the flow cytometry world is fairly widespread. If we're developing drugs that are likely to interfere with DNA synthesis or cell cycle division, we want to look at the DNA profile of cells. Clinically, in cancer diagnosis and prognosis, one of the ways to look at that is the DNA content of tumours or the proliferative compartment of tumours. Although I'm going to be mainly talking about mammalian cell analysis today, we can analyse the DNA in all sorts of species. So in fermentation processes, looking at yeast DNA cell cycle is very important. And in botany, looking at the DNA content of plant cells. And there are also some very important applications of cell sorting, which I'm not really going to talk about today, other than to say that it can be done. So we can, for example, sort sperm on the basis of their DNA content. We can separate X and Y bearing sperm because they have slightly different DNA contents. Or one thing that I will mention near the end is that we can sort on cell cycle position to regrow or produce cell clones. And this is a cartoon that many of you will be familiar with, an overview of the mammalian cell cycle. Cells that are not dividing are said to be in the G0 state, G0 phase of the cell cycle. When they gear up to start replicating, they don't change their DNA content, but they do enter the G1 phase of the cell cycle. They start producing some of the proteins that they're going to need to progress. When DNA synthesis does start, the cells are said to enter the S, or the synthetic phase of the cell cycle, and they'll continue there until they've doubled their DNA content and then enter the called G2 phase of the cell cycle. Then they'll enter mitosis and either produce daughter cells that go back to this G0 or quiescent stage, or will take another round of cell division. But thinking about it in simple terms of the amount of DNA that's in a cell, both the G1 and G0 stages of the cell cycle have the same amount of DNA. So if we stain them with a DNA binding dye, we'll see a population here. As cells enter S, DNA content is starting to increase. Therefore, they take up more DNA binding dye, they become more fluorescent, and they'll move to the right on that histogram. And then when they double their DNA, they'll have twice as much, they'll be twice as fluorescent, they'll appear twice as far up the DNA histogram. So here are our cells in G2 and mitosis. So this is one way of looking at the cell cycle, but there are other ways. I'm talking mainly of using single parameter DNA analysis today, which is great because it's applicable to very many cell types, particularly in the research environment, in the lab. We can look at DNA very quickly in cell cultures, but it can also be used in primary tissue. One slight disadvantage, and I won't talk too much about disadvantages today, but one slight disadvantage is that it doesn't tell us how many times cells have replicated. And in those cases, we might use one of these slightly more advanced techniques for looking for proliferation. One of those is looking at incorporation of BRDU, bromidioxyuridine, which allows us to follow cells through one or two cell divisions. If we want to look at more cells divisions than that, we can use what we call the BRDU Hertz quenching technique, which allows us to look at 3 or 4 cell divisions. But if we want to look at more than that, and sometimes we want to see a cell progressing through 5, 6, 7 divisions, then we would use a dye dilution technique, things like CFSE, the cell trace dyes that you may have heard of. But I'm going to be talking principally about this, looking at the DNA histogram alone. And how do we do that? I sort of intimated this already. We find a dye that binds to DNA. And there are very many of them out there. This is just a few examples. Some of them may be familiar to you, things like propylium the Herx dyes, DAPI. They all act in slightly different ways, and because flow cytometry is a fluorescence-based technique, we have to know a little bit about the fluorescence characteristics of these dyes. We need to know which laser wavelengths they're excited, and we need to know at which wavelengths they emit light. We may also want to know how they bind. So this immediately raises the question, which dye would I choose to look at DNA analysis in my cells? To some extent depends on your cytometer. It depends on the excitation wavelengths you have available. Pretty much all flow cytometers in the world will have a blue 488 nanometer laser light, which allows us to excite dyes like propylium iodide, 7AAD. Many flow cytometers will have a red laser around about 630, 640 nanometers. This allows us to excite dyes like topro-3. And if we have access to a UV or a violet line, this allows us to excite dyes like Herx and DAPI. It may depend on the specificity of the dye. Dyes like propylium iodide, they're small molecules, they intercalate into the helix without any real specificity. Whereas some dyes have a preference for particular base pair ratios. So Herx will bind to AT regions of DNA, whereas dyes like chromomycin will bind to GC-rich regions of DNA. That can be very important if you're looking at, for example, chromosome analysis, because the difference in base pair ratios allows you to separate different chromosome types. It may also depend on what you want to do with your cells. Do you need to keep your cells alive afterwards? The list I showed you on the previous slide, most of those dyes, in order to bind to DNA, require the cells to be permeable, either by fixation or by some sort of permeabilization agent, and therefore pretty much non-viable. So although you can analyze the DNA, you can't do too much with the cells afterwards. Of course, sometimes that's exactly what you want to do. There are only a few dyes that allow us to do this, that will stain the DNA in a live cell. Up until a few years ago, Herx 33342 was the only one of those. But this requires a UV or a violet laser, which weren't particularly common. Whereas more recently, dyes like TRAC5 and the dye cycle dyes have been developed to do the same function, to stain the DNA in live cells. But that's the minority, really, of DNA analysis. Normally, we're analysing the DNA in cells on fixed or permeabilised cells, And of course the advantage of our fixed cells means we can store our samples. We may also be looking at simultaneously other markers which may be within inside the cells. So we may need to fix the cells to get our antibodies into the cells. But sometimes the quality of the DNA is maybe not quite as good in a fixed cell as in a permeabilised cell. The disadvantage of permeabilisation means that we have more limited storage time. We really need to run those samples relatively quickly after we've prepared them. But because permeabilisation tends to strip either complete cytoplasm from the cell or removes a lot of cytoplasmic contents, we can sometimes get better resolved DNA peaks. So obviously the question, which fixation or permeabilisation reagent do we use? And in general, we can divide those into three areas. We can use alcohols, generally ethanol or methanol, and these are dehydrating, coagulating fixatives. They change the conformation of proteins, which is good for DNA because it means that all the histones and other associated proteins that are attached to DNA become coagulated, and it allows pretty good access of our DNA binding guides to the DNA. We can also use cross-linking fixatives, things like formaldehyde. These produce cross-links between proteins in the cell, so almost produce a cage of proteins around the DNA, which can make it more problematic for our DNA dye to get in. But sometimes that fixative is needed because, for example, you want to look at fluorescent protein expression at the same time. Occasionally we will use detergent methods, things like Triton, which is a very harsh detergent, or maybe saponin, which is a bit more lenient, and these will disrupt the plasma membrane and the nuclear membrane to allow access to our DNA binding dye. But whichever method we use, in an ideal world, what we'd like to see on our flow cytometer is something that looks like this. All of our cells that are in G1 should contain the same amount of DNA as any other cell that's in G1. So they should take up the same amount of DNA binding dye, they should fluoresce with exactly the same amount of fluorescence, and they should appear in a single channel on our flow cytometer. Cells that are in G2 have twice as much DNA, take up twice as much dye, become twice as fluorescent, and appear twice as far up the DNA histogram. And S-phase cells will be here somewhere in the middle. However, of course, we don't live in the ideal world, we live in the real world, and what we always see is a spread of data. Not all cells that are in G1 will take up exactly the same amount of DNA binding dye. So the profile that we see is determined by, yes, the total DNA content. It's also going to depend on some of our sample preparation and some of our instrument considerations as well. And all of these factors contribute to the spread of this data. But we judge the success of our staining for DNA by looking at the CV, the coefficient of variation of this G1 peak. So the narrower that peak, the lower the CV, the better the preparation, and the more easy it is to be able to delineate the different phases of the cell cycle. So what are the causes of heterogeneity within DNA histogram? Well, there are going to be some biological variations, particularly with cell lines. Cell lines tend to hang around in labs for many years, and they become heterogeneous in nature. They become a mixture of lots of different subclones, which can cause this biological variability and increase the CV of our DNA peak. There are some potential problems with our cytometer. Our lasers need to be very well aligned. We need to make sure that our cells are focused in the cytometer so that they all go to the laser beam at the same point. We'll have some sample preparation issues which may be related to fixation, and of course we also have to consider the amount of dye that we're going to put into our cells. So how does this manifest itself? So if we take a DNA histogram of a established cell line, we see something that looks like a recognizable G1, a G2 peak, and cells that are in S phase. But we see a reasonably big variation, big spread of our G1 peak. We can take these cells to a cell sorter and sort slices through this G1 peak. And if we grow up our cells, or if we take single cell clones and grow them up, what we can find is that we can tighten each of those peaks. And what we're doing in effect here is pulling out the different subclones in there. And we may not be able to get away from this, but if we have a cell line that has a very big, broad distribution and wide CV, we may be able to aid our experiments by doing some initial cloning. We need to make sure that our cytometer is behaving optimally. Now, if you're lucky enough to take your samples to a core facility, this is the sort of thing that will be done in the background. Hopefully it will be done by the core facility itself. What we want to try to ensure is that our stream through which our cells pass and our flow cytometer is optimally aligned to the laser, so that the cells always pass through the same point of the laser beam. That gives us a reasonably tight CV, which we can judge generally by running beads before we run out samples. If for any reason our laser has become misaligned, there's the potential for those cells to pass through a different part of the laser where there's maybe more variability in the light that's produced, and that increase in variability will manifest itself as an increase in CV. Even with a very well aligned machine, we have to think about how we pass the cells through the flow cytometer. Many cytometers allow you to adjust the pressure that you apply to your sample, and that has two consequences. First of all, it means you can pass more cells through the cytometer, which is great because it means you can get your data a little bit quicker, but there's a trade-off for that. As we increase our sample pressure, the core size within our flow cytometer, within our flow cell, increases. That means that there's more variability where each cell can pass through the laser beam. And that increase in variability causes an increase in CV. So for DNA analysis, where we're looking at relatively high resolution data, we want to make sure that we run our samples on the lowest pressure possible. And if we want to run them quickly, the best way to do that is to make sure our samples are as concentrated as possible. We can always dilute them if we find that we're going through too quickly. Sample preparation can be a problem as well. It's almost impossible to get a completely viable single cell suspension, whichever method we use. We're always going to have a few dead cells, we're always going to have a few clumps of cells together. And dead cells in any flow cytometry experiment can cause us problems. Particularly when we're looking at DNA analysis, dead cells start to degrade their DNA, so they're going to look like they've got less DNA than a normal cell. Dead cells will also release their DNA, and DNA is notoriously sticky, and that can cause clumping, something that we don't want in our flow cytometer. But we can target these cells. We can target dead cells by putting in a viability dye. So we've already implied that for most of our DNA analysis, we need our cells to be fixed or permeabilised, in which case they're all dead. But there are some dyes out there that we can add to our cells before we fix them that will mark the dead cells so that we can remove them later on. And these are quite often referred to as live dead fixable dyes, and that allows us to specifically target cells that were dead before we fix them. And fixation itself is a cause of variability. I mentioned that we quite often use either alcohols or aldehydes in our flow cytometry experiments. Alcohols are very good because they coagulate the proteins, they make the accessibility of the dye to the DNA very even, which improves our CVs. We don't get so much data spread. If, however, we use a formaldehyde fixation, because of the cross-linking nature of the fixative, it means there's more variability of how dye binds to our DNA, and we see an increase in the CV. We also need to think about the dye concentration that we use. The binding of the DNA dyes to DNA is in most cases not irreversible. So unless we're saturating our binding sites with the DNA dye, we can again see variability. And we can do a very quick experiment. We can take our cells at a given concentration and vary the concentration of dye that we use. In this case, we're looking at prapidium iodide. So we see as the concentration of dye goes up, the CV of our G1 peak comes down, and the staining intensity goes up. And by the time we get to around about 30 micrograms per mil, we're saturating. But of course, that's going to be cell concentration dependent. So if you change the concentration of your cells, that may no longer be saturating. But again, it's a cause of heterogeneity. Flow cytometry is a single cell technique. We have to make sure that we're analysing single cells. So if we think about what's happening within our cytometer again, as a cell enters the laser beam, we start getting fluorescence. Our fluorochromes are being hit by the photons from the laser, fluorescence is being emitted, we're detecting that in our photomultiplier tube, and we start to see a signal. As the cell enters the laser beam, when it's completely enclosed in the laser beam, we get that signal increases up to a maximum, and then as the cell exits the laser beam, that signal decays, and we get a pulse of light associated with each cell. In general, in most flow cytometers, we can measure three things. We can measure the height of that pulse, the pulse width, and the area under that curve. With DNA analysis, we can use those parameters to help us exclude cell doublets. Because if I plot pulse width against pulse area, my G1 cells will take a certain amount of time to go through the cytometer. S-phase cells are a little bit bigger, their nuclei are a little bit bigger, so their pulse width will start to increase. And the area under this curve will also increase because there's more DNA, we're getting more photons coming out. And our G2 and mitotic cells, again a little bit bigger, take a little bit longer to go through the beam than a G1 cell. The two cells that are stuck together will have a considerably bigger pulse width. So we can start again to target these cells and exclude them from our data analysis. So in practical terms, we can look at, in this case, pulse width, pulse area, and exclude our doublets that have a longer width. So if we look at our DNA histogram completely ungated, so we look at all the data, we can see a recognizable G1 peak, but this is much more difficult to interpret. Once we apply our gate, then we can see our G1, few S-phase cells and our G2 cells much more easily. And depending on the cytometer you use, there are different ways of excluding cell doublets. We tend to use either a pulse width versus pulse area of our DNA dye signal, or pulse height against pulse area. It doesn't matter which one you use, they're performing the same function. We're excluding doublets and clumps. Sometimes our causes of heterogeneity are again down to the instability of our cytometer. So one plot I find is very useful, either during acquisition of the data files or later on in analysis, is to plot the DNA signal against time. So time is pretty much a free parameter on flow cytometers. We know when we started making measurements, we know when we finished. We can plot that against any parameter we want. So as long as I see nice G1 peak remaining in about the same place, this plot is very even, there's no data dropout or anything like that, I'm quite happy that the machine is performing to the best of its ability. But if there's a problem, I can use time as a gate to exclude things that I'm not interested in. So if you've approached your DNA analysis in a sensible way, you've worked out what the best fixation to use, which is the best dye to use, which is going to be influenced by your cytometer, you need to set up the cytometer to make sure that you get the data that you're interested in. So the way I would normally do that is to set the following plots. I would normally use a plot, a forward and side scatter. This just really allows me to identify the cell population that I'm interested in. I'm going to want to try and exclude doublets. So in this case I'm using a height against area plot for my propylium iodide signal. I'm using PI in this example. I want to make sure I exclude dead cells. So before I fix these cells, I added amine reactive dye, which picks out cells that were dead before I fixed them. And I also like to have a plot of forward scatter against my DNA signal. This really allows me to help monitor again the cytometer performance. This is the G1 peak, this is our G2 peak. They should be fairly parallel to the forward scatter axis. If they're not, then that gives me an indication that maybe the cytometer isn't performing correctly and I may need to perform a clean or something like that. And I would adjust my voltage of my DNA channel to put the G1 peak at a fairly arbitrary point. I'm always going to have a control in my experiment, an untreated sample or an exponentially growing sample. So as long as I set the voltage correctly with that, I would leave it in the same place for the rest of my run. I tend to put the G1 peak around about a quarter of the entire scale. The reason I do that is because, particularly in a lot of cell lines, we see the duplication of DNA. So cells will move from a diploid state to tetraploid, and then rather than dividing and going back to diploid, they'll carry on dividing. And any octaploid or 8N peak would be about here, so it would be on scale. If I increase this peak so that it was about halfway up the axis, then any cells that are greater than G2 amount of DNA would be off the axis. We wouldn't be able to see them. And what I try to do is collect at least 10,000 relevant single events. So I haven't shown any gating on here, but during the machine setup, I normally just set a single gate here to look at my single cells. And I acquire 10 to 15,000 of those if I'm just interested in DNA content of the cells. And once I've acquired my data, then I'm a bit more rigorous with the data analysis. First of all, I would tend to start with this gate, the doublet discrimination gate, to make sure that I select only the single cells. Then within that population of single cells, I want to see how many are alive, how many are viable. Within that viable population of single cells, can I use scatter to exclude the cells that I think might be maybe dying, that haven't been picked up by this dye, or are looking a little odd. So I'll apply a scatter gate as well. And then finally I'll have my DNA histogram. In fact, the more gates you apply, this goes for any flow cytometry experiment, the more gates you apply, the more specific you can be about the population that you're interested in. So finally, I have a result, a DNA histogram, where I can define cells that are in G1, cells that are in S phase, cells that are in G2. And you'll note here I'm using a linear scale to acquire this data. I'm only seeing a doubling of fluorescence between cells in G2 and G1, so I don't need to use a logarithmic scale to compress that. I can quite easily do it on a linear scale. So once I've done this, I've optimized my technique, I can use it now to get some results, to ask some questions. I can compare, for example, proliferation between two different cell types or the same cell type after different treatments. So after leaving my cells in the incubator over the weekend, so serum starving them, I can compare how many cells are in G1, how many cells S, how many are in G2. I can also look at the effect of added drugs. So in this case, we've added a drug that should block our cells in S phase, and of course I'm always going to have a control, an untreated sample, and then I can compare my treated sample. I can see a nice increase of cells here in the S phase. Similarly, a mitotic block, untreated control, and then when we treat our cells with a mitotic blocker, we see a large number of cells piling up in that phase of the cell cycle. This is done on cultured cells. We can also look at DNA analysis of primary cells, and we do this clinically looking at tumours. So if we take a biopsy from a patient, we should see a normal DNA content. If we take a biopsy from a tumour cells will quite often change their DNA content. They'll become aneuploid. They won't have the normal 46 chromosome worth amount of DNA. And we can pick this up very nicely by single parameter DNA analysis in flow cytometry. So we can see here an aneuploid population. And by overlaying those two populations, we can see that the aneuploid population has what we call a DNA index or a diploid index of 1.36. That means that this aneuploid peak has about 36% more DNA than the normal diploid peak. And that number can sometimes give us important prognostic or diagnostic information. One disadvantage of single parameter DNA analysis is that how do we get the numbers? I can take any analysis program and I can put markers fairly subjectively around what I consider to be the G1, the S, and G2 peaks. But I'm always guessing a little bit where that G1 peak ends and where S begins. Where does S end and G2 begin? And this has always been a problem. We've known this from the very early days of flow cytometry. So what the community has come up with is a whole series of mathematical modelling programs that allow us to deconvolute the DNA histograms. Okay, so they'll fit Gaussian curves to the G1, the G2, and the S phase. And this makes it a little bit more objective, which is what we're aiming to do. And there are several algorithms and programs out there that do this for you. These are some examples of them. There's a module within Flojo and some of the other general flow cytometry analysis programs. But there are two specific programs, ModFit and Multicycle, that are designed especially for deconvoluting DNA histograms. So just briefly towards the end of this, I just wanted to come back to vital DNA staining or DNA on live cells, because there are some advantages of that and some specific applications. We don't need to fix the cells. Okay, so they're going to be live. We can do anything that we would normally do with a live cell. We can very easily exclude dead cells. We simply add any of these viability dyes to it. And we may want to use them when we're looking for fluorescent protein expression. The fluorescent protein that we've added may have some effect on cell cycles, so we may want to specifically target the GFP, RFP positive cells. Isolating stem cells by looking at the DNA content has been very popular using the so-called SP or side population assay. Sometimes we want to look at functional studies such as apoptosis, or we may want to reculture the cells. We want to sort them according to DNA content and go away and do something with those cells. And as I mentioned at the beginning, there are a limited number of dyes that allow us to do this. And with all of these dyes, though, the workflow is very similar. We can use the same sort of histograms. We can use a pulse height, a pulse area plot to exclude our doublets or include our singlet cells. We can exclude dead cells, and we can get a histogram very similar to the ones we just seen in propylium iodide. However, in terms of practical considerations, it's a little bit trickier. If you've got a mammalian cell line and you come to my lab, I would say if you fix those cells in ethanol and stain them with 50 micrograms per mil propylium iodide, you're going to get a pretty good profile every time. I can't say that as easily with these vital DNA dyes because different cells react in different ways. We may need to vary the concentration of dye that we add to our cells. We may need to vary the time that the dye is on there. We may also need to vary the temperature of incubation. Some dyes work better at room temperature than they do at 37. And also we don't want these dyes to be toxic because the reason we're using them is we want to make sure we can do something with the cells afterwards. So we want to make sure that they don't interfere with viability or functionality. So we tend to do a lot more preparation steps with the vital DNA dyes than we do when we're fixing cells. But if we do that, we can use it to do things that we can't do in any other way. So an example of where we've been sorting cells, we'll take a DNA profile of a cell line, which we believe there's a small tetraploid component. So basically if we sort cells in the diploid region and regrow them, recapitulate that picture. If we sort the few events that are here above the tetraploid peak and regrow them, what we end up producing is tetraploid clones. Okay? Of course we couldn't do that if the cells weren't viable. So that's been a relatively quick run through of some of the ways we use DNA analysis in flow cytometry. So just to summarise what I've been saying, DNA staining by whichever dye you choose to use is a very widespread technique. It's very widespread because of these two things. It's simple and it's versatile. We have lots of different DNA dyes we can use, so no matter which lasers and which optical filters you have on your cytometer, you'll nearly always find a dye that will suit your purposes. And as we've seen over the last 40, 50 years, while flow cytometry has become an indispensable technique, DNA analysis is very useful in all these sort of areas, looking at cell proliferation, cell growth, and using it to assess how drugs will interact with the cell cycle. So with that, I'd like to thank you for your attention, and if there are any questions, I will take those now. I'll hand back to Barry.
Thanks, Derek. That was an excellent presentation. So we have a few questions from the audience. If anyone else has any questions, please feel free to post them in the questions box that appears on the right of your screen. Derek, the first question we have is there a minimum number of lasers needed for a flow cytometer to be able to perform DNA analysis?
Well, you're only ever going to use one, really, for your DNA dye. So as long as you have the right laser for the dye you want to do, yes. Most of the early work that was done on cell cycle analysis was using propylene iodide because it was very widely available. And 20, 25 years ago, most cytometers only had a blue laser. So you only need one.
And just a dye that will be excited off that one, I guess.
Exactly, yes.
Yeah, and second one here is, which dye would you personally consider the gold standard?
Our gold standard technique here is ethanol fixation cells and propylene iodide. Some people seem to prefer to use DAPI. I think that does depend on the setup that you have, because as well as having the right laser wavelength, the signal you get from your DNA dye also depends on things like the strength of the laser, the power of the laser. Sometimes it's easier to vary that with say, UV lasers that used to be attached to sorters.
Okay, it's going to be determined, I guess, what facility and what works in your setup.
So the major advantage for us of fixing an ethanol propylene iodide is it's so easy.
The next question, Derek, is how many events do you think you should acquire per sample?
It's not so much the events you acquire per sample, but the number of events that you're going to be interested in after you've gated and worked out. So, if we were to say you always acquire 10,000 cells, if 90% of your cells were dead before you fix them, you're going to exclude those from your analysis. You'll only end up with 1,000 relevant cells, which is sort of towards the low end for statistical robustness. So I would normally set up the cytometer to make sure that I acquire somewhere between 10 and 20,000 events. Obviously, sometimes that depends on the number of cells you have, but not total events, relevant events. So I'd set my singlet gate, potentially set the viability gate as well. Use those as prerequisite before I start acquiring data.
You spoke about saturating concentrations of dyes. Do you find this is common to most cell types when they're fixed, or is there a lot of variation between different cell types?
If you always stick to the same cell concentration when you're dealing with mammalian cells, there's not a lot of variability. But of course, stoichiometry depends not only on dye concentration, but cell concentration as well. So if you've done your titration and worked out that you saturate at a certain micrograms on 1 million cells per mil, and then you try and do 20 million cells per mil in your samples, yes, you may be out of that range. It always pays to try and keep the cell concentration constant.
Question here, is there an approach that's good to separate cells within G2 phase and M phase?
It depends what you want to do. Yes, we can specifically target mitotic cells by using antibodies against proteins that are specifically expressed in mitosis. So we use an antiphospho H3 protein antibody quite a lot, which phospho H3 is specifically phosphorylated in mitosis. But in order to detect that, we need to fix the cells. So we can get a percentage, we can get a mitotic index. We couldn't do anything with those cells afterwards.
Is it possible to analyze condensed chromosomes with PI, for example, where colchicine is used in karyotyping? So I guess, can we use PI to actually analyze chromosomes?
With PI alone, it's quite difficult. And I think if you look in the literature, there are attempts at looking at chromatin condensation, and you normally use glycoacridine orange to do that, because you'll get slightly different signals depending on how it binds to the DNA. It's quite a tricky technique, I think, to get to work. But so something like rubidium iodide is a bit unspecific in the way it binds. Okay, so part of the reason why you get a spread of the data around G1 and G2 is to do with chromatin condensation, because not all cells in G1 have their chromatin in exactly the same conformation. That leads to this data spread. To target that specifically it is possible, but I think you'd have to spend a little bit of time working that up.
Okay, great. Our next question is, how would you best optimize the vital dye concentration, time and temperature of incubation prior to the experiment? So I guess question about optimization.
Yes, I mean that's I suppose relatively straightforward. You'd set up the same sort of thing you'd set up for titration of an antibody. So we'd look at we'd set up various tubes with different concentration of the dye, incubate them at the appropriate temperature, whether that's room temperature or 37, and sample at different time points. Okay, so we would check then a couple of things. We would check the quality of the DNA histogram, so at what point is our CV optimal. But we would also check the viability, because we want to make sure that we're getting a good CV, but we're keeping the cells alive, that we're not killing them. But even then, that may not be enough, because we may come to a point where we think that's a good concentration of dye, then it's not toxic, but it's not toxic at that time point, is it? You want to try and leave those cells in culture for 24 hours. Do they proliferate? Do they maintain that viability?
Great. And the next question, Derek, just to clarify that forward and side scatter as well as single fluorescence is required for cell cycle analysis to exclude doublets. So I guess, do we use the forward and side scatter also to exclude our doublets?
I would use as many things that I have at my disposal to exclude doublets. So as well as looking at the area and height of the DNA signal, which is a very specific signal, scatter's not as quantitative, it's a bit of a qualitative parameter on a flow cytometer. And so I tend not to use that as a primary gate. But we can also look at the area, the height and the width of our scatter parameters as well. And it's always useful to acquire those, to have them clicked in your software, because sometimes they will pick up a few more doublets, exclude a few more. And if you're looking for very subtle changes, you really want to be as sure as you can be that in your final gate you're looking at single whole cells.
Next question. So can you choose CD14 monocytes gated, and then on DNA histograms on this selected cell population for G0 and C1 and G2 DNA levels, I guess that's maybe G1 and G2 DNA levels, with and without culturing mixed leukocyte cultures with standard media and a proliferation agent added to cultures? So I guess maybe it's, can we do I guess various different tests on the one sample?
Yes, I mean, this is the beauty of flow cytometry, isn't it? That we can, as long as we've got the fluorochromes available to us, we can add as many as our flow cytometer will let us. So it may be that you have to change the way you look at the DNA content, for example. If you're looking at a surface-stained molecule, something like CD14, dyes like FITC, for example, will survive ethanol fixation very well. So we can stain our cells with surface marker fixing ethanol and do our DNA staining, as we talked about with the iodide. If we find that we can't do that, then we might have to think of using a different approach. We might use a formaldehyde fixation, which means our DNA profile may not be quite as good, but it would keep the surface staining intact. Or maybe if that doesn't work, we would use then maybe a vital dye, where we would stain our cells but not have to fix them and get that information. But as long as we can define the populations that we're interested in, this is what flow cytometry is good at. Within that population, we can look at whatever else that we've added into our mix. If DNA content is one of those, we can define our monocytes and their CD14 expression and look at the histogram of those alone rather than the whole population.
Yeah, I think the high speed and multi-parametric availability on the cytometry is really what sets it apart and really gives it its power. So that brings us to the end of the seminar. So I'd like to thank you again, Derek, for a very illuminating presentation and a great questions discussion. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com, and it should be available within the next 24 hours. There you can also see the other webinars we've lined up for you in Bitesize Bio's Webinar Festival. So until next time, good luck in your research and goodbye from all of us at Bitesize Bio.